इसमें क्या करना है?
Hello, Dr. Devjani.
Hello, sir. Hello.
Good evening.
Good evening.
Dr. Devjani, our director, has joined.
Hello, Dr. Devjani.
Hello, sir. Hello, sir.
How are you?
Fine, fine. Dr. Rashmi, we'll start sharp at 3 because that was
already 3.
3. Okay. Okay. Tell Dr. Burman then. Dr. Thimanna. Dr. Thimanna.
Yes, ma'am. Background.
கணபதி இது எப்படி ரெக்கார்ட் பண்றது?
ಹಾ ಓಕೆ ಸರ್ ಮೇಡಮ್ ಒಂದ್ಸರಿ ಫೋನ್ ಮಾಡಿ ಹೇಳಿ ಮೇಡಮ್ ವೇಟ್ ಮೇಡಮ್ ಇದು ನಿಮಗೆ ಫೋನ್ ಮಾಡ್ತಿದ್ದಾರೆ Ma'am, shall we start?
Should we start, Dr. Devjani? You are muted.
Yes, we can start. Our
Okay.
director sir is there.
Already
He has joined, no?
Yes,
So,
already joined. Yes.
Sir, should we start then?
Okay, ma'am.
Please go ahead.
Okay. Okay, sir.
Okay. Good afternoon to all the dignitaries, distinguished guests, ladies and gentlemen. I welcome you all to 13th Dr. S. Pradhan Memorial Lecture. Let's begin today's function by invocating Goddess Saraswati and by paying floral tributes to late Dr. S. Pradhan, sir.
जय जय हे भगवति सुरभारति तव चरणं प्रणमाम्यहम्। जय जय हे भगवति सुरभारति तव चरणं प्रणमाम्यहम्। नाद ब्रह्ममयि जय वागेश्वरि नाद ब्रह्ममयि जय वागेश्वरि चरणं ते गच्छाम्यहम्। जय जय हे भगवति सुरभारति तव चरणं प्रणमाम्यहम्। त्वमसि शरण्या त्रिभुवनधन्या त्वमसि शरण्या त्रिभुवनधन्या सुरमुनिवन्दितचरणा नवरसमधुरा कवितामुखरा नवरसमधुरा कवितामुखरा स्मितरुचिरुचिराभरणा जय जय हे भगवति सुरभारति तव चरणं प्रणमाम्यहम्। आशिना भव मानसहंसे आशिना भव मानसहंसे कुञ्जतुहिना शशिधवाले हरजरदां कुरु बुद्धिविकासं हरजरदां कुरु बुद्धिविकासं सितपङ्कजतनु विमले जय जय हे भगवति सुरभारति तव चरणं प्रणमाम्यहम्। नाद ब्रह्ममयि जय वागेश्वरि नाद ब्रह्ममयि जय वागेश्वरि चरणं ते गच्छाम्यहम्। जय जय हे भगवति सुरभारति तव चरणं प्रणमाम्यहम् 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 प्रणमाम्यहम्।
Philosophical Society of India.
Thank you, everyone. May I request Dr. Debjani Dey ma'am, convener of 13th Dr. S. Pradhan Memorial Lecture and Head Division of Entomology, to welcome the dignitaries and participants and also brief about Pradhan Memorial Lecture Series.
A very good afternoon to all. On behalf of the students, scientists, staff of the Division of Entomology, ICR IARI, I welcome our Honorable Director and Vice Chancellor IARI, Dr. A.K. Singh, and request him to chair the session. I also welcome the esteemed speaker of the day, a world-renowned entomologist, Dr. Zedar Khan. A very warm welcome to our beloved Dean, and Joint Director Education, Dr. Rashmi Agarwal, Madam, and Joint Director Extension, Dr. Toma, and Dr. Balraj Singh, Project Coordinator, Honeybees, and also our former colleague, Dr. Subhash Chandra, currently Director in CIPM. I also welcome another illustrious alumnus of Division of Entomology, Dr. Aravind Kumar Pradhan, IAS, and currently Country Director of ICRISAT for this memorial lecture. A warm welcome to the present and former directors of various ICR Institute, especially Dr. Varghese and Dr. Chandish Balal, Madam, former directors of NBAIR, professors and scientists and heads of various divisions of IARI, faculty members from various colleges of University of Delhi, HU, AMU, various SAUs, scientists from other ICAR institutes, former heads, professors, scientists, and retired members of our entomology family, office bearers of Entomological Society of India, I welcome you all. And finally, a very warm and special welcome to the daughters of late Dr. S. Pradhan, Mrs. Shalini and Mrs. Ragini, their spouses, children, grandchildren, and many other family members who have taken time off to join this function. Again, I welcome one and all for the prestigious 13th Dr. S. Pradhan Memorial Lecture. The lecture is extremely relevant to the current times due to the onslaught of several invasive pests into our country. And Dr. Zedar Khan, a world-renowned entomologist who has been working on control of many of these important lepidopterans, has also agreed to interact with the students and scientists. And all of us who have queries can use the chat box. And also students and scientists desiring to interact with the speaker can remain logged in after the vote of thanks. Dr. S. Pradhan, as you all know, is an entomologist. During his 33 years of professional career, made such an impact on entomological research and teaching that entomology and plant protection science came to the forefront of agricultural research in our country. His success story would continue to encourage many generations of plant protection scientists of the country. He joined the Indian, then Imperial Agriculture Research Institute in 1940, became the first professor of Division of Entomology in 1958, and thereafter the head of the division in 1962, and continued till February 5th, 1973, when he left this mortal world. Dr. Pradhan was the first to visualize the country's need for integrated pest management instead of the conventional method of chemical control alone, which, though successful at the time, had started effects. He emphasized on the fact that protection research was more needed than production research in tropics, and therefore crop protection research should have its rightful place in the overall agricultural research efforts of the country. His forceful arguments for adopting integrated pest management to fully realize the production potential of new high-yielding varieties of different crops had their impact, and the country adopted IPM as a national strategy, but it was only sadly after his demise. To remember his contributions to Indian entomology, a corpus fund was put in place with the contributions of his family members, individual entomologists, ex-students, and his well-wishers. As part of the Golden Jubilee year of our country's independence, Division of Entomology started Dr. S. Pradhan Memorial Lecture Series. Till date, 12 lectures have been delivered by eminent entomologists in this prestigious lecture series on various aspects of entomology. An attempt has been made to bring forth some of his most significant contributions in various spheres of fundamental and applied entomological research in a documentary which will follow soon. Scientists of the division, especially Dr. Timanna, Dr. Sagar, and Dr. Sashank, with their experience of scripting and editing Pusa Samachar, have improved the earlier version of the documentary immensely. Now, please, the documentary to follow. Thank you.
Dr. Shyam Sundar Lal Pradhan had a humble beginning. He was born on May 13, 1913 at village Diva in Baharish district of Uttar Pradesh. His father, Sri Guru Prasad Pradhan, was a village level officer of the state government, having 5 sons and 3 daughters. After initial studies at a nearby village school, Pradhan passed high school examination of UP board in 1928 with distinction in mathematics. He passed intermediate examination with a gold medal from Radhaswamy Educational Institute at Agra in 1930. Thereafter, he joined Lucknow University for his graduation and post-graduation. Later, he joined as a research scholar in the same university under eminent zoologist Professor K.N. Bal. He worked on functional morphology of insects for his DSc degree in 1938. He carried out part of his PhD research work at Rothamsted Experimental Station, United Kingdom, and was awarded PhD from the same university. Dr. Pradhan started his career at Indian Council of Agricultural Research at Gorakhpur. Soon after, he joined Indian Agricultural Research Institute as assistant entomologist at the institute substation at Karnal, where he did his famous research on insect ecology. Then he joined Division of Entomology in 1948 and established country's first school of insect toxicology. Dr. Pradhan became the first professor of entomology in 1958 and head of the division in 1962 and continued till 1973. Dr. Pradhan was a visionary and scientist par excellence. With versatile interests, he contributed significantly to various aspects of fundamental and applied entomological research. He was the first to identify and describe natal glands in Coleopterans, coiling and uncoiling mechanism of proboscis in Lepidoptera, homology of male genitalia, and regeneration of midgut epithelium in insects. Having deep understanding of insect crop environment interactions, proficiency in mathematical derivations, he derived 2 mathematical equations, popularly called as Pradhan's equations, which explain relationship between temperature and insect development. Based on these equations, he developed biometer and biograph. These are a ready reckoner for correlating insect development with temperature fluctuations. Dr. Pradhan was a pioneer in insect toxicology research and demonstrated strong anti-feeding activity of neem. He was the first to report insecticide resistance development in Singara beetle to DDT and BHC. Under his leadership, Division of Entomology developed a cheap, scientifically fabricated and effective grain storage structure, popularly called as Pusa bin, and later larger version Pusa cubicle. He propounded the famous biotic theory on the periodicity of locust cycles, which explained the distribution and abundance of locusts both in time and space. Dr. Pradhan was a gifted teacher and guided the research work of 65 students in the discipline of entomology. Dr. Pradhan's scientific papers as well as popular articles bore his characteristic clarity of thought and acumen of presenting complex subjects in a simple form. 2 of his books, Insect Pests of Crops and Agricultural Entomology and Pest Control, would always remain valuable to agricultural scientists. Besides heading the Division of Entomology for more than a decade, Dr. Pradhan was a founder member and also President of Entomological Society of India for 4 terms. The two positions and his towering personality made him the national leader of the country's crop protection specialists. The society took great strides under his stewardship. The membership of the society increased and the Indian Journal of Entomology grew in circulation and stature. Dr. Pradhan was regarded as an authority on insect ecology, toxicology, and integrated pest control, not only in India but internationally. In one such recognition, he was invited by UNESCO as one of the selected 6 authorities to write different chapters in the book on human and animal ecology. He was a member of FAO panel of experts on integrated pest management and chaired important sessions of international and national symposia and conferences. Dr. Pradhan was chairman of Entomology Committee of ICAR for a number of years and guided the planning and implementation of national policies on entomological research, teaching and extension. For his outstanding contributions, Dr. Pradhan was posthumously awarded Dr. P.B. Sarkar Endowment Prize for the triennium 1971 to 1974. Indian scientists would always remain inspired by Dr. Pradhan's work, who was rightly called father of modern applied entomology by late Professor K.N. Mehrotra, ex-head Division of Entomology, ICAR, IARI. In his memory, Division of Entomology has been organizing series of lectures since 1998 to till date.
Thank you, Debjani ma'am, for welcoming our guests and briefing about the Pradhan Memorial Lecture Series. I also thank our audiovisual team for the wonderful documentary. Now, may I request Dr. Rashmi Agarwal ma'am, Dean and Joint Director Education, to introduce the chairman and speaker of 13th Dr. S. Pradhan Memorial Lecture Series.
Thank you, Bhagishri. Good afternoon, everyone. Distinguished guests who have joined today 13th Dr. S. Pradhan Memorial Lecture Award Series. Our Honorable Director, Dr. A.K. Singh, who is the Chief Guest and Chairman and the Chief Patron of today's function. Eminent Speaker, Dr. Z.R. Rahman. All other faculty members of Division of Entomology, including Head of the Division, Dr. Debjani Dey. Students, ladies and gentlemen. It's my really pleasure and privilege to introduce briefly about the chairman of today's function, Dr. A.K. Singh. All of us know about Dr. A.K. Singh, who is a distinguished rice breeder and a distinguished alumnus of IRI. And he's better known as Basmati man, and a large number of Basmati varieties which he has bred has really brought prosperity to many of the Basmati farmers. Our distinguished speaker of today is Dr. Professor Zayoor Rahman Khan, who is again one of the distinguished alumni of IRI, who did his MSc and PhD both from IRI in the Department of Entomology. And immediately after doing his PhD, I will briefly introduce to you about the eminent speaker today. And after doing his PhD from IRI, he briefly joined Rajendra Agricultural University, Bihar in India. And thereafter, he did his postdoctoral from International Rice Research Institute, Philippines. And then he also joined as a research associate in Wisconsin, Madison, and again as a visiting senior scientist at Kansas State University from 1991 to 1993, before joining his present position as principal scientist and program leader at International Center of Insect Physiology and Ecology, Nairobi in Kenya, and he is there since 1993. He is also adjunct professor of entomology with Cornell University, USA. During his students' days when he was at IRI, Dr. Khan was very much impressed by Dr. Pradhan's vision on countries' need for integrated pest management. Instead of conventional chemical control alone, he went ahead and explored the many facets of insect-plant interactions during his PhD program. He has dedicated more than 30 years of his career for advancing the science and practice of entomology by studying and applying chemical ecology, insect behavior, insect and plant interactions, and also plant-soil interactions. Dr. Khan is internationally lauded for his development and Africa-wide dissemination of the innovative system of push-pull farming, which simultaneously addresses the issues of crop pests, soil improvement, food security, climate change, and sustainability. His work in developing the push-pull technology is a wonderful example demonstrating that creativity and innovation in agricultural sciences can provide practical solutions for the real problems of poor smallholders by removing constraints of their farming and ultimately leading to the better food security and sustainable agriculture and livelihood. He was awarded the Louis Melassis International Scientific Prize for his outstanding career in agriculture in 2015 for his exemplary and promising contribution in promoting innovation through research, development, and capacity building in agricultural system, as well as contributing towards addressing food security and poverty reduction. He is also honorary fellow of the Royal Entomological Society, London, fellow of Entomological Society of America, fellow of African Academy of Sciences. Then he is also council member, International Congress of Entomology, and president of International Branch of Entomological Society of America in 2019. And Dr. Khan has also authored more than 170 scientific papers and 20 books and book chapters. So it's today we have a very eminent speaker amongst us. And I welcome Dr. Khan, the speaker, and all distinguished guests who have joined today's 13th Dr. S. Pradhan Memorial Lecture. So I would like to invite our distinguished speaker to deliver his lecture.
Thank you very much, Dr. Agarwal, for a very kind introduction. I'm also very grateful for IRI, Department of Entomology, for inviting me. It is a great honor. I'm feeling like I'm back at IRI. If COVID would have not been there, I would have certainly come to deliver this lecture personally. And although I never met with Dr. Pradhan myself, because I joined IRI in 1975, and he had already passed away, but we heard a lot, and students used to discuss, our seniors used to discuss about Dr. Pradhan and the contribution he made, and we used to read his books, and that was very inspiring for almost all the scientists which came out from, not only from IRI, but from India. But when I moved to other parts of the world, people still knew Dr. Pradhan and his contributions of in Indian crop protection and Indian agriculture. So it was a great honor to know him, although not personally, but to know his work and talk about him at various platforms. So it was very nice that I was invited to deliver this lecture. And it was very surprising when I received a call from Dr. Day. I was in the US in August that I have to give a lecture, and I immediately accepted it. So before any delay, I would like to go and start my slides. Should I do that? Can I do that? Yes, thank you very much. Can you see that? Hello?
Yes, we can. Please go ahead.
Okay. Thank you very much. The title of my talk, as I was introduced, exploiting chemical ecology and plant signaling for developing sustainable crop protection strategies for a smallholder farmer. And this is a scientific journey of an Indian scientist from lab to land. I can see back very clearly how I started, and I thought to be a scientist, and how I became a field scientist from a lab scientist, and how I helped many farmers in improving their food security. I can't move without dedicating my talk to Dr. Pradhan, who was a world-renowned entomologist and dedicated his career for agricultural innovations and development. And when I became a fellow of Royal Entomological Society, I looked at the list of the fellows and I found him as a fellow, and it was a great honor to be associate myself with him through that fellowship. And he was also president of Entomological Society of India. My initial work when I started in Kenya was funded by some British donors, and I worked with scientists from Rotham Estate, from where Dr. Pradhan did his PhD. So that was also a very good link which I had with Dr. Pradhan. And this is, when I was preparing my lecture, I found some very old photographs of my association with IRI. And this is a photograph on my on left-hand side, where Dr. Pradhan, Dr. Pant, who became head after that, is standing with some of the students, and this is myself. And these are two other colleagues, Dr. Ramesh and Dr. Sarode. We were standing with Dr. Pant. And Dr. Pant left for UK right after we joined as master student. But he told us, we all wanted to do our PhD in toxicology, because that time everybody wanted to become toxicologist, because there are a lot of jobs. But he told us, go for some basic science. Like he told me that I should go and do my MSc with in insect taxonomy. So I was allocated with Dr. Faruqi to do my master's, whereas after my master's, I went and worked with Dr. Agarwal, who I worked with on insect-plant interactions. And this was also a very memorable picture, where all the professors and the scientists and the students were as assembled on one of the occasions. I think the occasion was probably one of the scientists was retiring. So these are very memorable photos, and I can say that my journey to become a scientist, it started at IRI, because that was my aim, to become a scientist, and inspiring from the work of Dr. Pradhan and Dr. Pant and other scientists, Dr. Agarwal, Dr. Mehrotra, Dr. Bhatia. We were seeing the work they were doing and how they were interacting with the farmers. That gave us inspiration to be a scientist, and then that was the start of the journey. After that, as Dr. Agarwal said, that I went to work as assistant professor at Rajendra Agriculture University, which is now a central university. At that time, it was not. I was there for 3 years, then went and worked with Dr. R.C. Saxena, who also delivered one of the first Pradhan lectures. So I went and worked with Dr. Saxena as a postdoctoral fellow at IRI. Dr. Saxena was a scientist there at the time when I went there. And my work was mainly on the leaf hoppers and plant hoppers. And I was trying to find out their feeding behavior. And the feeding behavior was very important in determining the resistance. And I developed some of the new methods like electronic feeding behavior, and some of these where you could use different dyes to find out whether the insect is feeding on xylem and phloem. And some of the papers I published from there were in very high demand. Many scientists and virologists used my work very widely in the world. Then from there, after finishing, I moved to University of Wisconsin at Madison, where I worked with Professor Dale Norris, who was a world-famous insect-plant interaction scientist. And I worked on the cabbage looper and the resistance in soybean and published a couple of papers. From there, I went back to Erie and I joined as a scientist at Erie, the International Rice Research Institute, and I worked on Lepidopteran pest, stem borers, and leaf folders, and also wrote two books. One was World Bibliography of Rice Stem Borers and Insect Pest of Rice with Dr. M.D. Pathak. M.D. Pathak was also alumni of IRI Entomology Department and who was the Director of Research at IRI. Being entomologist, I worked together with him on many projects, but this was one of the projects of Insect Pest of Rice, which book has become quite popular. Even the World Bibliography of Rice Stem Borers is very popular. It's very widely used by the rice entomologist, still being used by rice entomologists. After finishing there, I went and worked at Kansas State University for 2 years on wheat Hessian fly, and in the Department of Entomology under the chairmanship of Professor Michael Smith, and also wrote a book there, which was Techniques for Evaluating Insect Resistance in Crop Plants, which was co-authored by Michael Smith, myself, and Dr. M.D. Pathak. And that book has become quite popular. It is still being taught to graduate students all over the world. It is one of the very few books on the techniques. From there, in 1993, I joined ECP, which is called International Center of Insect Physiology and Ecology. It was established in 1970, and it was founded by a Kenyan entomologist who was educated at Cambridge, Professor Thomas Odhiambo. And ECP's mission was to help alleviate poverty, ensure food security, and improve overall health, but not applying pesticides, but by understanding the chemical ecology and ecology of the insects and plants, and to do the capacity building and to see that how can we have environmentally safe pest management strategies, pest and vector management strategies. So it has a very wide mission. The center, it has 4 programs. It has plant health, environmental health, animal health, and human health. And the scientists are working in these 4 departments. I work in the plant health department. And this is Thomas Odhiambo, who was a Cambridge-educated professor at University of Nairobi, and then he established this international center in 1970. And when I joined ECP in 1993, he challenged me to understand the science of mixed cropping. Why the farmers plant like beans and maize, or soybeans and maize together, and what are the chemical interactions between the 2 crops. And he also challenged me that how the stem borers come back on the maize and come to maize, and after maize is harvested, where they go and how they come back again. So that was a kind of my service contract, that I should start working. He gave me a 5-year contract. In most of the centers, we don't have lifelong appointments, but he gave me a 5-year contract and told me to start working and look for donor. And because we don't have funding from the organization, very big funding, but we have to look for donor and write a proposal. So I started doing that and looking at. So what came to, with all that experience which I had at Erie and the 2 US universities, I thought that now I am at a center where I have a long-term appointment and I must apply the chemical ecology. And chemical ecology was a fancy term at that time, where many scientists were trying to understand what happens when insect attacks a plant, how the natural enemies come and help the plant, and what happens in the root system, how the plants defend themselves against the weeds, and how they defend themselves against the insect. So that complex understanding and to identify the semiochemicals. So that was something which I thought that I must start working on it, and also try to understand the plant signaling. Plant signaling is very important. Many plants signal each other. If a plant has attacked by the pest, that plant will signal the other plant and tell that plant that the insect infestation has started, so the other plant can prepare itself. So the interaction is not only above ground, but the interaction is also underground. And the underground communication is through mycorrhizal networks. So we need to understand all that to have a successful strategy for crop protection. So my first insect to work was stem borers, and the stem borers, there are about 6 species of stem borers in Africa, but I started working with 2 when I came because they were very serious. And one was Chylopatellus, which came to Africa from Asia in 1930s and got established here. And the other one was a African stem borer, Busuela fusca, which is also very serious, and they can destroy the whole maize farming, and the farmers apply pesticide to control them. And if we estimate the loss of even 15%, the total maize in Africa, the loss is $1.5 billion, which is a very, very big economy of Africa. So one day I was traveling to the field, and I found that there was a small plot of maize which was planted here, and next to it was wild grasses. So I started doing the sampling, trying to find the stem borers which were associated with maize. But for curiosity, I also went and did some sampling here, and I found that the same stem borers are also here, but in much smaller numbers. Although both of them have the same family, but the question came, why maize cannot defend itself against the insect attack, and how the grasses can defend themselves. So that was a question. I went back to my lab with that question, and then I started reading and found that Africa is very diverse in the grasses, and many grasses have developed very smart strategies to defend themselves, because if they don't know how to defend themselves, the insects will destroy them. And there are also some literature that when they are attacked, they can also signal to the neighboring plants, and many wild grasses are also refugia for natural enemies. As you see here, the natural enemies are here. So it is very complex system, and we must understand that before developing any strategy to control stem borers in Africa. So what I did, that I started a survey of wild grasses, and we found that many of these grasses are associated with many of the stem borers. These stem borers are also present on the maize, but they are also present on these grasses. I was not an agronomist. I was not a grass specialist, so we had to interact with agronomists and collaborate with them and try to identify them. They did not know about insects. I knew about insects, but they knew about the grasses. So a lot of time went talking to them and trying to understand them, the grasses, to identify them, and established some nurseries of the grasses on my own research station. But also there was a lot of dissections which we undertook, of both on the maize and grasses, which required a lot of labor to see what species of stem borers are associated with grasses and with the maize. And that gave a very interesting understanding that that what species of stem borers what species of stem borers, these are the stem borers, Kylo, Buciolas, and other stem borers, are associated with which species of the grass. And as you can see that some of the grasses are associated with all the species of grasshoppers, and some of them are only with one or two species of with the grasses. So that was very important understanding and discovery, which we published very early, still in 1997. And it was very important discovery that we found a grass which is Napier grass. In India, we call it elephant grass. And the Napier grass, which is a perennial fodder, we found that many varieties of Napier grass were highly preferred by the stem borer egg laying as compared to maize. When you give a choice of Napier grass and maize plant, the stem borers prefer to lay egg on the Napier grass as compared to maize, but very few of them were able to develop from larvae to adulthood. That means the development of the stem borer on the Napier grass was highly reduced. At that time, we did not know why, but later we found that when the eggs are laid and larvae penetrate into the stem, the plant produces a gummy substance, and the small first and second star larvae get trapped in these gummy substances. We also looked into the development, and we found that in maize about 60 to 70% of the larvae become adult, but on the Napier grasses, only 5 to 10% become adult. So they die, and this is a mechanism which nature has provided to the plant to defend itself, and this is not present in the maize. So, and that's why the maize is heavily attacked by the stem borer. But now the question is how to defend it. What should we do? We also found out that Napier grass has a special mechanism when it becomes dark in the evening, and when the stem borer females become active, it starts producing some chemicals, the green volatile chemicals, which are highly attractive to maize stem borer females. And maize does not produce in that large quantity. And that's why the Napier grass becomes more attractive to stem borers for egg laying as compared to maize. So although there's large number of eggs are produced, are laid, very few are able to become, are able to grow and become adult on the Napier grass. So I thought that this would be a very good trap plant, by if you plant it, and it is a very good fodder. So if you plant it, there could be large number of eggs will be laid on the Napier grass. The other plant I came across accidentally was molasses grass, Melinis minutiflora. It is also an African grass, and it has a unique property. It repels the stem borer. If you plant it in between maize plants, the stem borer does not like to come and lay egg on the maize plant. But it has a property that it attracts the natural enemies. The natural enemies are attracted by this plant, and there are very few plants which do that without being damaged. And this plant is one of the plants which is can be used in the crop protection strategies. And in 1997, when we found this, that was a very big discovery. And the discovery was so big that when we submitted the paper in Nature, Nature accepted it. And we showed the chemicals which this plant produces. And these are the chemicals: ocimene, caryophyllene, nonatrine, humulene, and terpinolene. And these are the SOS chemicals which maize plant produces only after being attacked by the insect. But this plant is producing it in nature and attracting the natural enemies. So we thought that can we use it as a repellent plant and also a plant which could bring the natural enemies in the field. Later we discovered that molasses grass, when it is planted next to maize, without touching the leaf, without interacting the root, it gives the signal to maize plant. And the maize plant also starts doing the same thing which molasses grass does. And it starts attracting the natural enemies and repelling the stem borers. And this was, we later published in 2019 this paper through a PhD student. And this was a perfect example how plants can signal their neighboring plants or how plants can learn from each other. So we are still working on it to find the molecular basis of plant-to-plant signaling and interactions. But that discovery was enough now to start something in the field, because this was money we had got was from a donor, and the donor wanted us to show that you must go from lab to land now, work with the farmers and see that the farmers can accept to grow this Napier grass as an attractant and molasses grass as a repellent, and develop a technology which can control the stem borers. And I was not a social scientist. Now I can do that, but now at that time I was not. So I had to work with a social scientist from the national program, and the social scientist used to invite farmers. But he told me that the men and women should be invited separately, because the men will not speak in the presence of the men. So let's know from them. And most of the farmers in East Africa are women. So let's discuss with them whether they are comfortable in growing Napier grass and molasses grass, and have a technology which could control the stem borer. So we defined the term push-pull. Pull is the Napier grass, which is on the border. It is pulling the stem borer, and push is the molasses grass, which is here. It is pushing the insect, and after every 10 rows of maize, 1 row of molasses grass was planted. And this was a very initial stage of push-pull, where the stem borers were repelled from the maize by the smell or signaling which is coming from the molasses grass, and they are attracted to the napier grass, and napier grass does not allow its development. So in the field, we planted on the station as well as on the farmers' fields, and the farmers themselves did the counting, and some of them, average of 70%, there was a reduction in the stem borer damage. And that gave us a lot of pleasure. So what is push-pull? It is was at that time a novel approach. There are many people are now trying to develop the push-pull, and by understanding the mechanism of that govern the chemical ecology of plants and insects and plant signaling. These two are very important keywords, and we have to carefully select plants, and the plants have to be useful to the farmer. If you bring a plant which is not useful to the farmer, they will not accept the technology. But here both the plants were good fodder, and you plant both attractant and repellent plants and try to reduce the pest damage. So many farmers adopted it, and they were. This is KARI, is Kenya Agriculture Research Institute. We worked with them, and many farmers took it. They made a group, and they were quite happy with the technology. And many farmers, these are some of the early adopters. I will come back and talk about Lillian and John Gombe later, who was the first farmer who adopted. And Lillian wanted to have a cow in her household, and cow cannot come without fodder. And she found some good fodder now with the napier grass. And this farmer, Peter, already got a calf, and he adopted the technology. You can see the smile on his face. So we thought that we have the technology which is working, but it was not working, because we went to a region where there was another problem. There was a weed. The first region where we had success, the parasitic weed, which is called striga, was not there. Only a stem borer was a problem, and we were able to control it. But when we moved, we saw that the other regions, striga weed was most common. So these farmers did not want to accept the technology, because they say that this parasitic striga weed, as you see here, it is a parasitic weed which attaches itself with the root system of maize and sorghum. It is much more dangerous pest, and the women spend a lot of their time in weeding, but still the maize cannot produce anything. So that was an eye-opener for us, that you can develop a technology for one pest, but it cannot be acceptable in other regions because the other pest could be more important. And those farmers told us that even if you are able to control the stem borer, a striga weed is going to kill our crop. And what we saw, that 25% of African land is infested with a striga, where maize is grown. And the total loss is $2.5 billion, which is more than a stem borer. So, but we were not weed scientists, we were all entomologists. So we had no answer to the farmer, but accidentally discovered a plant which is called Desmodium. The Desmodium is a fodder legume, and we found that after a lot of experiment, I can't go through all the experiment in the limited time, but when the Desmodium is intercropped with the maize, it not only repelled the stem borer, but also controlled a striga completely. You can see here on this side where maize mono is planted, the striga weed has completely devastated this maize. Here there is we have grown with Desmodium, is a fodder legume, it has controlled Striga. So this was one of the biggest discovery which we had in 1998, as early as 1998. And I compared with many other legumes, beans, green gram, cowpea, desmodium, and found that desmodium was the best to control the Striga beet. So then tested this in an experiment where desmodium is planted here, and the distilled water passed through the root system, and the root exudates came through here, and both these pots had Striga seed with it, with the maize. And here there is no desmodium, and you can see here where there is no desmodium, the Striga which was present here has already dwarfed and later killed the maize. But when the desmodium was planted, it controlled the Striga. Only one or two Striga emerged, and that showed how strong was the chemistry of the root system of desmodium in controlling Striga. And that gave us all the strength to go back to the farmers to show them that this plant can be planted, and you can control both stem borer and Striga. You can control both this pest through one cropping system. So later we also identified the chemistry to find out how the root system, what chemicals are produced by the root system of desmodium, the legume, which controls Striga. And that was very interesting chemistry, which we published first in phytochemistry, then later we have published now several papers describing each of those chemicals and how they can control Striga. So now we had another push-pull. We replaced the molasses grass with desmodium, which is a legume, and napier grass remained the same. So now we have this new version of push-pull. And new push-pull has these two perennial plants, the desmodium, which was repelling the stem borers and controlling the Striga, and and the napier grass attracted and killed the stem borers. Desmodium being perennial and a legume, it also improved the soil. So that was the advantage, and both of them provided fodder to the cows, because most of the farmer had either the dairy goats or dairy cows, and they could keep on harvesting the fodder from napier grass and desmodium and feed their cattle. So the farmers who had a very poor land in this region or other regions where desmodium was present, they were not able to control. They were applying a lot of weedicides, and they did not have money. They were not able to do that. Can have a very nice crop without any weedicide, as you see here, and their yield was improved several times. So that was this lady was one of the first in this region where the Striga was a problem, and still planting after more than 22 years, still planting the push-pull system, and was also on the Swiss radio, Swiss television. She was featured on Swiss TV, which was very nice to her and to other farmers. Soon, when we published this work, and Swiss TV broadcasted, National Geographic came and talked to us and published something in National Geographic in 1999. That gave a very big boost to us. But soon it was followed by the New Scientist in 1998 and 2001. There were two articles, Farmer's Friend and An Ordinary Miracle. And these two were published by the New Scientist. They gave us further boost, and many more funding agencies were attracted to the work. And then the Nature published Making Crops Cry for Help. That was for the how when plant is attacked by the insect, how it releases the chemical to call for help. So these were some very important articles written by others about my work, and it was very good to see that how this can attract more research. And soon the ambassador of United States, Bushnell, came to see the work. And you can see I'm standing here. This was the Director General of ECP, and this was scientific advisor to the ambassador. They all came, and I was explaining. She was very happy, and the US also funded. And after her, the other ambassador, Johnny Carson, also came to see the work, and I'm showing the work in the field. And Johnny Carson was also quite pleased with what he saw in the field. So we continued working with it. We understood the chemical ecology of the push and pull plants, and also what is happening in the root system, that how the desmodium is controlling the striga, what chemicals it is producing. So we continued working with the Rothamsted research, or before it was called Rothamsted Experimental Station, and later it changed the name Rothamsted Research, and we continued working with the lab of Professor John Pickett. Push-pull technology had great benefits by controlling the stem borer, reducing the striga, and reducing the improving the yields 2 to 3 times. But also when we did the economics, it has a greater return to the labor and greater return to the gross, and had a much better gross benefits. Farmers who planted the push-pull had a lot of fodder in the term of napier grass and desmodium, and they were able to have the cows and goats, and were able to improve the fodder, and not only fodder but also the milk production between 2 to 3 times in their household. And more than 150,000 farmers in East Africa were able to integrate the dairy cows with the push-pull system. So we started looking into dissemination pathways and see that how the farmers learn the technology so that we can improve its dissemination to the farmers. And what we found that in Africa, where there are many tribes, they learn in different ways. Many farmers learn from each other, early adopters. Many learn from farmer teachers. Many needs a field day to show them. Many listen to the radios. The others don't listen to the radio. Some like the extension staff. Some don't interact with the extension staff. So we tried to understand them and develop a strategy how to deploy the best dissemination path. Many of them were able to read, but many of them were not able to read. The farmer field days were conducted. Farmer teachers were very important to teach them. Radio program, TV program, now the participatory videos, and farmer field school. We applied all of them in the region where the farmers were benefited the most. And this was very important, which was farmer teacher. We gave them a bicycle, and this is the Lord Sainsbury. Lord Sainsbury funded us initially when we started the work, and he was also Minister of Science of UK. So he came to meet with the farmer teachers and to see that how the work was going on, and he was very pleased and funded more to continue the work. But while we were trying to improve it and take it further, what happened that there was a climate change, and the climate change was so severe that our push-pull could not, the companion plants which were perennial could not survive longer. So now we went to European Union and described them about the work, and asked to develop a new funding to develop a new push pull, which could be called climate smart push pull. And European Union funded us initially 3 million euros and then later 5 million euros. And we discovered new plants. We discovered bracharia grass and this is a new desmodium, which is called green leaf desmodium. We combined them, and they were drought tolerant, and they were able to survive very long droughts of Africa, and but also helped to increase the production a couple of times, 3 to 4 times, and control the stem borer and the striga weed. All these papers have some publication. As you see here, we published, most of these things are published. And bracharia also has early herbivore alert. And early herbivore alert is when the stem borers laid egg there. It recognized that the eggs are laid even before the larvae emerged and start feeding, and started attracting the natural enemies to come and help. So that's why we called it early herbivore alert. And this is very good in improving the biological control. Later, we found that there are varieties of maize which are also have early herbivore alert, and it can also control. It can signal each other, and it can also defend itself against the pest damage. So that work is still going on. We discovered first in 2011 and published in Ecology Letters. But this is a very important work for us, where we are trying to identify the genetic mechanism of early herbivore alert. So this is climate smart push pull, where we use the bracharia and the green leaf desmodium. We replaced the early desmodium, which was silver leaf desmodium, to green leaf, and Napier grass was replaced with desmodium, with bracharia, because these are more drought tolerant plant. But soon we realized that there are problems, and the problems were red spider mite, which attacked the bracharia, and the green leaf desmodium, which we selected, was not able to flower near the equator. So that was a problem. We started new research, and this is the red spider mite, which was attacking our trap plant, as you can see here, which made the farmers very unhappy. Soon we had a visit by the ambassador of European Union, and he came to meet with the farmers, and we showed him the problems, and the ambassador funded these are all disabled farmers who had adopted the push pull. So he met with them, and then he funded us new money to have a new push pull, to select new plants which could have resistance to these red spider mites. So we started new research at that time, where we selected plants which were resistant, also had farmers' perception, and found the plants which were not attacked by red spider mites, which was called Xeris, but was a good trap plant for stem borers, and had bigger biomass, more drought tolerance, and also liked by the farmers. And also got some highly drought tolerant desmodium, which could flower near the equator and produce the seed. So farmers can have their own seeds rather than buying seed from the market, and also have a very good mechanism of control of striga. So this is Desmodium incanum, which could flower and produce the seed, also control the striga. So we developed a new push-pull, which we call a third generation push-pull, like 3G in the phone. So 3G, third generation push-pull, which does exactly the same thing, but it is resistant to red spider mite and Desmodium flowers. And like this is what it is planted in the field. And now this is our new generation of push-pull, which we are giving to the farmers. And it has very good performance for control of stem borers, fall armyworm, and the yield production. And when we did a farmer's perception we found that the farmers preferred it as compared to the other conventional push pull. While we were all doing that, the fall armyworm invaded Africa in 2016-17. Before going to India, it invaded Africa, and now the problem was what to do with the push pull, because we were hearing that it can destroy more than 100 plants. So can it destroy the Napier grass and Bracharia and Desmodium? And what is going to happen now to the push pull? While we were thinking that, you have seen this photo before, Lillian and John Bagombe, who adopted the push pull in 1997. I received a call in 2017, exactly 20 years after, saying that Professor Khan, the push-pull technology which you have planted here, I have expanded it, and none of my field is attacked by the fall armyworm. So it looks like the Desmodium and the Napier grass is controlling the fall armyworm. I visited them after 20 years. They had been changed quite a bit, and they still had the push-pull. Not only had push-pull, they had expanded it into 4 fields, and they showed. And they also showed me some of the neighbors' plot where there was no push-pull. The maize plant was totally devastated by fall armyworm, but where there was push-pull, the maize was protected against the push-pull. We immediately went to European Union again and described them about this new problem, which was fall armyworm, and there was a visit by 3 people, Bernard Ray, Myra, and Bathome, these 3 people, and they came and looked at the work which we are doing and demonstrated that how the push-pull can control the fall armyworm, and we got some new money to start the work on the push-pull and fall armyworm control from European Union. And in a survey we demonstrated, which was also published, that how the push-pull can reduce the fall armyworm damage from almost 100% to 20% or less. But also compared with other cropping systems using bean, soybean, groundnut, other things. So both the climate smart and conventional push-pull had a better control of fall armyworm. Now fall armyworm has invaded in 2018 to Asia, China, and everywhere. So we were asked to write an article that push-pull farming system controls fall armyworm and lesson from Africa. So we wrote that, and we had a meeting with FAO in Bangkok, and there was a lot of requests that how can we bring the push-pull system to Asia. So we are doing that, trying to do that, and also to South America, how to take the push-pull to South America. So we are still screening the grasses, because in push-pull system, we found that it is Desmodium which repels the fall armyworm, but the grass, which is a trap plant, we have no trap plant which can control the fall armyworm, which can trap the fall armyworm. So we are selecting the new plants, and we have found some which we have just published in 2021, and the work is continuing. But the chemistry, we are also looking at the chemistry of the Desmodium and how it repels the fall armyworm. We also showed that fall armyworm can. We also showed that in addition to stem borer and fall armyworm, the push-pull also controls aflatoxin in the maize, and that work is also going on. We also started some soil fertility work because it is a legume, and we found the farmers which have planted the push-pull for almost 20 years, and we looked at the soil fertility, and we found that it improves carbon, phosphorus, and nitrogen, And not only that, but it also improves the organic matter in the field. Recently, we have integrated the push pull with the vegetables, and because there was a demand, and we found that desmodium, which is planted in between here, as you see, not only can repel the cereal pest, but it's also repelling the vegetable pest. And we have just started this work and shown that how this could be integrated, and the demand of the push pull has increased a lot. The farmers can not only intercrop with the maize, but in the off season they can where there is no maize, they can plant vegetables in between desmodium. And these are the farmers who have planted it, and they're describing how the system is working and how they are benefiting from this. So if you look at overall benefits, the overall benefits are stem borer, armyworm, striga control, improvement in fodder and milk production, climate change adaptation, climate change mitigation, soil fertility improvement, aflatoxin control, and control of vegetable pest now, which we started in 2020. And it aligns itself, the technology, with various UN Sustainable Development Goals. It aligns with reduced poverty, zero hunger, gender, education, and climate action, the soil fertility, and several others. So it aligns with itself with UN development goals, and it is also mentioned as a chapter in a recent FAO book, the whole chapter on the push-pull. But very interestingly, it was mentioned by UN Secretary General in the UN Assembly report in 2015, where you see here the push-pull has been described as a technology which can help millions of poor in Africa and beyond. So this is a report from the UN General Assembly. And the New York Times has put my name here as a true deserver of World Food Prize. I have not received it, but it was the New York Times has recommended it, that because of the person who developed the push-pull technology should be given the World Food Prize. And 2010, there was a visit of K.A. Acharya, who is the president of Forum of Environmental Journalists in India. She came with some environmental journalists from the world to see the push-pull, and she published an article in Hindu. But before doing that, she went and talked to Dr. Swaminathan about the push-pull, and Dr. Swaminathan had already known because he had invited me to give a talk in his center. And so his comment was that push-pull is a perfect example of a low-cost, low-input production system, providing a pathway to an evergreen revolution without causing environmental and social harm. The technology ensures both soil health and crop security and can be adapted to different growing conditions. So it can also benefit farmers beyond Africa. So that was the statement from Dr. Swaminathan, which made me very proud. And it was published in Hindu. And the article was written by K. Acharya, who was a president of Forum of Environmental Journalists. So to conclude, I don't want to take a lot of time. To conclude, it is a kind of technology which is necessary to support the green revolution in Africa, because African farmers can't afford fertilizer and pesticide. They're poor, and the rainfall is erratic. They don't have irrigation. So they need a system which can bring them the green revolution, because they missed the green revolution. And the new discoveries which we had is control of aflatoxin, fall armyworm, soil health improvement, climate change mitigation, These are great opportunities for further enhancing the effectiveness of push-pull technology. But the integration of vegetables, which we started doing last year, and other high-value crops to intensify the push-pull system will make it very attractive. It's making it very attractive to youth. And what is happening that the youth in Africa and maybe in also Asia, they migrate to cities and out of Africa. Here they are moving out of Africa. They are lining up to go to Europe. And if we could provide them a good income on their own farm by giving the technologies which can give them at least $5 to $6 return to the labor in a day or a little more, they will probably not move out of their land, and they can they will continue farming on their land. So what lesson we learn is science for society. The science we do should be for society, and society and what society hopes of science, because all the money we receive from donors, whether in India or abroad, comes from comes from the taxpayers' money, and that benefit should go back to those people in improving their livelihood, and that is very, very important. So that should be our aim to work. This work I started in 1993, and I'm still continuing it. I can see it has been more than 28 years. I'm doing it. So and I think about the smallholder farmers, so that they can be benefited from the work I'm doing. I get feedback from them and try to look for the new ways how to help them. Thank you very much.
Thank you very much, Z.R. Khan sir, for the enlightening lecture and inspiring young researchers on chemical ecology by narrating your work with very informative facts and figures. Sir, it's really great listening to you. May I request our Honorable Director, IRI, and Chairman of 13th Pradhan Memorial Lecture, Dr. A.K. Singh sir, to release the booklet of the lecture and to give chairman remarks.
Okay, let me share the presentation here. All right, so I'll just scroll it down in this mode. This is the compilation of the presentation. And this is available online on the IRI website. So I'm sure you're all interested in what Dr. Khan has presented. Very nice lecture. You can visit the site and you can see the entire presentation and also about Dr. Khan. Thank you so much. Well, friends, it has been a great lecture and truly mesmerizing. The one thing is doing good science for the benefit of the society, and at the same time making efforts to take the science to society. And from basic research to applied research to communication with the farmers, you could see all the three components of agriculture research, education, extension system in the presentation by Dr. Khan. It has been wonderful, and I would request each one of you to join me in giving a big round of applause to Dr. Khan for his excellent presentation. His simplicity of presentation is something truly admiring. We must also learn how to communicate science. Communicating science has been a big casualty in many places, and unless we do that, we will not touch the heart of common people. And that's what Dr. Khan has been able to do. That also, when you are doing something starting from say 90s, even before that, the documentation that he has done, starting from his IRI days to Pusa, Bihar, back to IRI, different postdoc positions, then working at Kenya, this present center, the perfect documentation, and therefore the story is so lucid that you don't lose the track and you understand clearly what he wanted to communicate. Now, this is strategy of chemical ecology, the communication of plant to plant, plant to insect, the entire environment is something which is very interesting. I think he made a mention in his initial remarks that we need to understand the chemical ecology of mixed cropping system. Mixed cropping system has been in practice, he described most examples taking some weed, whether it was Desmodium or other weed species, and use them for controlling insect or weed management. But in mixed cropping system, I remember in our childhood a 5-crop mixture, that is pigeon pea, then urd bean, tama, that is small millet, sesame, jowar, and these 5-crop mixture was a common farming practice during Kharif season, and it extended till Rabi season. What kind of communication happens between these cultivated species? We probably don't understand much in terms of chemical language, what Dr. Khan has described in relation to weeds and what are those important chemicals which are produced for communication and how we can harvest this benefit. There are many weed associated problem. For example, in case of mustard, we have got Arbanki. Arbanki is a major menace. And if you go to many areas in Rajasthan, we find even chemical control is not very effective. If we can find some component of pull and push strategy which can manage Arbanki, that would be really wonderful. Likewise, in terms of insecticide usage, if you see, few crops like cotton and rice, they are the 2 crops, and of course after that vegetables which consume almost put together 90% of total insecticides. In case of rice, for example, if we think of developing some pull and push strategy, one has to look at also the economics of pull and push strategy. How much cropped area will go to pull crop, how much will go to push crop, and how much will go to main crop. And overall, what is the economics of this cultivation is something very important to look at, to adopt it as a strategy in a large area, particularly in the crops where the maximum consumption of insecticide, pesticide is happening. So from Dr. Khan's lecture, I would like our entomology team to kindly take lead. And I think we are not working in this particular area as far as my knowledge goes. We do have some primary knowledge. For example, Dr. Sidhu is there, and he would say that if you use marigold, it will act as an attractant, and some of the other crops which are used as an intercrop to, attract the crops and use them as a trap crop and so on. But understanding the chemical aspect of it, understanding the genetics of it, understanding the molecular biology of it is something which can form the core of research component in years to come. Because this is one of the most sustainable way of managing the biotic stresses, whether it is insect pests, diseases, or weeds, which is equally applicable to all these components. Yesterday only I was in Gorakhpur interacting with some farmers, some rice farmers, and some farmers came to me and said they had the problem of stem borer that he was referring to. And in an aromatic rice variety, which is more prone to kala namak, which is highly aromatic, is highly sensitive to stem borer. And they wanted the control. And the moment I said, Pradhan, Keldan, Sanvax, and Coragen, and so on and so forth, all these chemical, the farmer said, sir, जब हम इसको खेत में डालते हैं ग्रेन्यूल, छिड़कने वाली दवा तो ठीक है, लेकिन जब ही हम ग्रेन्यूल खेत में डालते हैं तो हमारे खेत के सारे केंचुए जो है मर गए और दिखते नहीं है, जो कि पहले खेत में भरे होते थे. तो अगर आप सजेस्ट कर रहे हैं तो हमको कोई छिड़कने वाली दवा बताइए, ग्रेन्यूल तो मत बताइए। ये हमारी हाथ जोड़ के प्रार्थना। अब आप देखिए कि गांव में एक किसान की ये सोच है कि जो हम तरीका अपना रहे हैं बड़े पैमाने पर, उसमें सस्टेनेबिलिटी का बहुत बड़ा एस्पेक्ट है, क्योंकि हम बार बार कहते हैं कि सॉयल इज अ लिविंग एंटिटी, और उसमें अगर इतना ज्यादा हम पेस्टिसाइड डालेंगे तो इसका इम्पैक्ट जो है लॉन्ग रन में ठीक नहीं होगा। एक हमको जुडिशियस कॉम्बिनेशंस बैलेंस बना करके चलना है। तो आई थिंक दिस इज न्यू एरिया व्हिच डॉक्टर खान हैज presented his views, his findings, which is widely recognized, as you could see from his presentation. I would like that we should make some initiative in this direction, at least in agronomy division. There are at least 5 to 10 scientists in agronomy division. They are working on cropping system, intercropping system, different forms of intercropping system. Can we take up some of these aspects in intercropping system? What is happening in terms of the chemical ecology of the cultivation system of farming practices? That would be really very, very interesting. And you see, it's not that you can't make good publication, and Dr. Khan is one of the best example. He has shown his paper published in Nature from studying, but provided you must understand and identify a right kind of problem and address it from a right angle. So I would like to congratulate Dr. Khan for sparing time being with us. Unless you, we can have, if you have some time, maybe if there are some questions, we can take. We had very distinguished guest with us, although in memorial lecture generally we don't have, but I would invite, if there are some questions. This is a very interesting topic. We had Dr. Padi with us, Professor T.V. Singh from Telangana University, and many senior people who have joined. If there is any question or comment from any one of you, please feel free before we close.
Yes, Dr. Khan, there is a question from Dr. Sushil Saxena. He wants to know, does the push-pull technology work for pests of cereals and millet crops only? What about the borders of cotton, sugarcane, and okra? Does this technology work against sucking pests of horticultural crops?
Oh, sorry. Yeah. It is a very good question. It can work on almost everything, as long as you identify the chemicals which can attract and repel, and that is very time-consuming work. Like we have started now with kales and onion, and some of the tomatoes, to see that if desmodium can repel the pests, and what crops can attract the pest. So it is something which you need to identify the chemicals which are a strong attractant and a strong repellent. I have not done anything on the sucking insect yet. Most of my work is on the Lepidopterans, where they can find a host and they can lay their eggs, and they can be repelled, and the natural enemies can be attracted. But it will not be difficult also to work on the leaf hoppers and plant hoppers of the sucking insects. Since we don't have very important sucking pests here, my work has not expanded to that area. But it should be possible. We should be able to work on that.
Madam Chandesh from she wanted to know, what is your take on the published research on the use of Fusarium oxysporum strigoi for management of striga? This be integrated with the push-pull strategy?
Yes, there is some published work to control the striga. And we have seen some of the field trials in that, and the results are still a mixed result. But the farmers, they have to buy a product to use it. fusarium from, they have to go to the shop and use it. Whereas in the use of the desmodium to control the striga, you don't have to buy anything. Once you buy the desmodium seed and plant it one time, because these are perennial plants, it will keep on providing the control year after year, year after year. And it will also provide the fodder for the cow, because the desmodium is a very high protein fodder. So the farmers like to harvest and give it to the cattle, to cows and dairy cows and the goats, and improve the milk production. So it does several things, and farmers like a technology which can do several things, not only control the one aspect like a striga. Here it controls the striga, stem borer, fall armyworm, and also it provides the fodder and improves the soil fertility. So, so when we compare this with fusarium, fusarium only controls striga weed. So, and desmodium controls in such a way, there is a perfect control. We don't have to add anything in the farm. And why to use more money? But those farmers who don't have desmodium, they can try it. But I have seen very mixed result. The result has been very mixed from place to place.
Dr. Khan, just one query in this context. What is the mechanism of desmodium interaction with the striga? How does it control?
Okay, the desmodium root produces two different chemicals, two different group of chemicals. One group of chemical, it stimulates the striga seed to germinate. So it is a pseudo germination chemical. So it germinates, but the other group of chemical, it immediately come in and it stops its attachment with the root system. So it is a very novel mechanism which we published in phytochemistry, and then later we published in several journals. And we looked into many other legumes, and most of the legume has only one mechanism, that is the germination stimulant, but they don't have inhibition. There is no chemical which inhibits its attachment. So that's why.
Orvanki is also a parasite on mustard. Will
yes.
it work, desmodium on Orvanki?
It will work. We have started some work in Sudan.
Okay.
With Orbanki and some of the vegetables, and with some PhD student. But now Sudan is in turmoil, so I am not receiving the result from the field experiments. But there was a very good control. Initial work in the pots was shown that if desmodium was planted, Orbanki was not able to attach itself with the beans, because there's the beans. So you could have a student who could look at it, the Orbanki's interaction with desmodium, and it has the same mechanism with Orbanki. I went to Sudan and I looked at Orbanki, so it should work.
Okay, thank you.
Yeah.
I think, sir, you have already answered this question. There is a question. Is there any push-pull strategy for management of pot borers in pulse crop?
You need to develop. We don't have a ready-made thing which is available in the shelf. Since it is a new area, people develop like cotton. We are also trying to see can we develop something for cotton, for the pot borer. The bollworm of the cotton. So we are trying to see this, because it takes time. It is not 1 or 2 day or 1 3-year experiment. It takes time. You have to select the plants, the best plant. So this is a long term, and Indian scientists are best placed to do that, because you have the whole career you can spend on the same crop looking into, whereas we in the international centers, we don't have a long-term contract. We have 2-year, 3-year contracts, and also the funding has to come from outside. Sometimes the donors get tired of funding the same thing; they don't fund, so people change the area. But national programs like India, which is very richly funded, you could take this kind of work and continue doing that. And collaboration is very important. The collaboration with chemical ecologists and agronomists, social scientists. So that collaboration has to be built, and it has to be worked. Like, I'm not a chemical ecologist. I did not come as a chemical ecologist. I worked with Rothamsted. I learned from them. Social science, I was not a social scientist, but I started interacting with social science, scientists, and then, learned from them how to do it. Even agronomy, I learned from them. So we need to have a very wide collaboration, and it has to be a long-term work.
Sir, one last question from Akola. The professor is writing, we have a serious problem of Cuscuta on soybean. Will desmodium work against Cuscuta? He should try. He should try intercropping desmodium, and it should work with Cuscuta. It should work. It is just to try. But I can answer any question. I'm available now. I will be available on email. Anyone, you please distribute my email address. I will be able to answer. A few months ago, I delivered a talk to China. They are still asking the questions. I keep on sending them the publication, answering. So it will be a great pleasure to send the papers, publications, and answer the question of any student, any scientist, anyone who wants to interact with me.
Thank you, Ashok. Thank you so much.
Thank you so much.
Yeah.
Thank you, Zedar Khan sir, for addressing our audience. May I request AK Singh sir to felicitate the man of today's program, Dr. Zedar Khan sir, with a memorable memento.
So I can only show you from here, and this will be presented to you and sent to you later. This is a memento with a picture of Dr. Pradhan.
Thank you very much.
Yeah, this is for you.
Thank you. Thank you very much. Thank you. It's a great honor.
Also, it's all, and that will be sent to you. Thank you, sir.
Thank you. I will be very pleased to accept them. I could have come personally myself, but I have not been to India for almost 2 years now. So I would have come personally, but everything is so restricted. Travel. Yeah.
That would have been wonderful, but we look forward for another opportunity when we'll have.
Thank you. Thank you very much. And I make sure when I come, I generally come to IRI to meet with some colleagues. So I do always do that. Yeah.
Thank you, sir. May I request Dr. Rashmi Agarwal ma'am, Dean and Joint Director Education, to honor Zedar Khan sir with a certificate.
Sir, it's my pleasure and an honor, sir. So this certificate also we will give you, sir. Either we will post or when you come to IRI. We look forward to meet you, sir, personally. Excellent lecture.
Thank you very much. Thank you very much, ma'am.
Congratulations, sir.
Thank you.
Congratulations, Zedar Khan sir. May I request Dr. Mukesh K. Dillon sir, Principal Scientist, Division of Entomology, to propose a formal vote of thanks.
Thank you, Bhagirishi. Most Honorable Dr. A.K. Singh, Director, IRI, Dr. Professor Zedar Khan, world-renowned entomologist, respected family members of Dr. S. Pradhan, dignitaries, distinguished guests from across length and breadth of the country, joint directors of IRI, heads of divisions of IRI, professors of different faculties of IRI, former entomologists, colleagues, scientists, staff, ladies and gentlemen. A very good afternoon to all of you. On behalf of the 13th S Memorial Lecture, S Pradhan Memorial Lecture Committee, and on my own behalf, I thank Dr. A.K. Singh, Patron and Director, IRI, for his wholehearted support and guidance in successful organization of this program, and also sparing his valuable time for attending this function. I owe special gratitude to Dr. Zedar Khan an accomplished and world-renowned entomologist, for accepting our invitation, sparing his valuable time and excellent presentation of the 13th HES Memorial Lecture. I had an opportunity to interact with Dr. Khan at various occasions, like during international congresses of entomology in South Korea at Daegu in 2012, Orlando in Florida in 2016, and we had very nice interaction during those times. We are grateful to Dr. Rasmi Agrawal, Dean and Dr. B.S. Tomar, Joint Director Extension, Dr. Subhash Chandra, Director NCIPM, Dr. Balraj Singh, Project Coordinator, AICRP on Honeybees and Pollinators, Dr. Arvind Padhi, Country Director, ICRISAT, former directors of various ICR institutes, other dignitaries and invited guests from various ICR institutes, SAUs, and other universities like DU, BHU, AMU, heads of divisions of IRI, professors of different faculties of IRI, office bearers of Entomological Society of India, invitees, scientists of IRI, former entomologist colleagues for sparing their valuable time and gracing the occasion. I thank the most important guests today, Mrs. Ragini and Mrs. Shalini, their spouses, grandchildren, and relatives of Dr. S. Pradhan for their presence and gracing the occasion. Special thanks to Dr. R. Burman, Principal Scientist, Division of Agriculture Extension, for providing online Zoom platform. And also thank the Central Photo Lab for helping in making this documentary. Thanks and congratulations to Dr. Devjani Day, convener of this 13th Dr. S. Pradhan Memorial Lecture and Head Division of Entomology, organizing committee members, and entomologist colleagues of the division for their wholehearted support and untiring efforts from the day of inception to final and successful organization of this program. I'll be failing in my duty if I don't thank the members and subcommittees, scientists, technical and office staff of Division of Entomology for extending all help and support for successful organization of this program. Once again, on behalf of the organizing committee and on behalf of IRI and on my own behalf, I once again thank you to all for gracing the occasion. Now I request all of you to please stand up at your places for national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता, पंजाब सिंधु गुजरात मराठा द्राविड़ उत्कल गंगा, विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा, तब शुभ नामे जागे तब शुभ आशीष मांगे, गाहे तब जय गाथा। जन गण मंगल गायक जय हे भारत भाग्य विधाता, जय हे जय हे जय हे जय 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 हे।
Thank you all. Now probably the students or other interested colleagues who wants to interact one-to-one with Dr. Khan, we are back to informal interaction with Dr. Khan.
Students,
I think students have to read first what they can ask to Dr. Khan.
Okay. No, I'm available here. It is only Going to be 2:25 in the afternoon here, so I still have a very long time here to sit in the office.
Sir, your mail ID is there in your lecture. So
Yes.
I suppose many will be just contacting you.
By mail is fine.
Yeah.
I can answer. Yeah, I can
Madam
answer. Please, sir.
Yes. Yes, yes, yes. Yes. Any questions? Maybe she is having connectivity issues. No. I think she is from Assam. So I think connectivity issues are there. Madam Balal has written that desmodium seeds are not easily available. She was trying to procure to do some experiment on push-pull. Colleague was trying.
Okay, I think you should look into gene bank. The gene bank will have it.
Definitely they'll have.
IRI gene bank will have all the
NBPGR will have.
Yeah, NBPGR will have the gene bank will have. So you need a small quantity and you can multiply. Yeah.
Chandis madam is
online. Yeah.
Yes, madam.
Sir, thank you so much, sir. Such an inspiring talk. We truly benefited, sir. Actually, we had an issue. We contacted various organizations. This was year before last when I was in service, and Dr. Amala was the person who was trying. And now she is herself growing the with whatever little bit of seeds she got from, I think, the Fodder Research Institute. She got some from Dharwad. And with that, she's herself trying to generate, and she's got some small. I was just messaging her and asking her. So she said she's now managed. But then initially it was a challenge, sir, to get the seeds. So it was a challenge, sir.
Yeah. Also, the seed companies, you might ask if they are having desmodium, because it is a very good fodder. And many large-scale dairy companies, they use it for the fodder for the cattle. Okay. So that's how I got it through some seed companies who had imported it for the dairy farmers. So that was the first time I got it through them. So that should be possible.
Okay. Thank you, sir. Thank you.
Yes, Dr. K.L. Srivastav, you want to say something? Of course, you have your old student here giving the talk. So if you want to ask anything, sir, you are welcome.
Actually, I wanted to ask Dr. Khan that whether this method and could be transferred to the IRI also.
Yes,
it can be transferred, and there was a time when Punjab Agricultural University transferred it. They used the napier grass, the elephant grass, to control the stem borers, and there were several publications from PAU. So it can work in India. So if PAU can take it, maybe IRI can also do that.
Thank you.
I think it will create a lot of, in fact, interest, because fall armyworm is really playing havoc in our institute itself. It is coming on all the crops.
Yeah.
Last season, it was a big problem.
Yes.
So somebody or the other will now take up surely this.
Sure.
One person, he's writing whether using pheromone traps and oviposition lures any good in incorporating into the push-pull strategy, because that is another way of attraction. So,
it is possible to use any method. It can be incorporated, because we call push-pull as a platform technology, and on the platform you can add anything you want. which is more useful. It can be valuable for that area. So if they find that they can use pheromone to attract and kill, that is fine. That can be done.
I want to ask some questions. Sushil sir.
Hello, Dr. Khan. Am I audible?
Yes.
I want to ask one question. In Africa, there are many countries which are cotton producing countries.
Yes.
Why the push-pull technology has not been found successful against the cotton borers in those countries?
People have not started working on it because we are looking into cotton now more seriously. And cotton has also got an advantage that its roots produce estragol, which also depletes the astraga seed. So where there is astraga, it is very useful to plant the cotton. But we have demonstrated that both desmodium and molasses grass can reduce the bollworm on the cotton, significant reduction in bollworm. But we have not yet found a trap plant yet, which farmers can use. Although there are several trap plants which have been reported in the literature, but we don't want to do it on the cost of the cereal production. You see, so we want to see if it can be done in rotation, cereal cotton rotation or something like that, where farmers can have a cereal crop and then they can have a commercial crop. So we are working towards it, and we might have something. Once we have something which is working, we will give to farmers which could use it. Yeah.
Thank you.
Any questions? Dr. Bhagyashree, who is working on vegetables mainly, and also Dr. Sagar, if you have some questions for Dr. Khan.
That's what the question was, only the question which has already been asked for the pod borers, madam. So that's already we need to work upon. Them, that's true.
Yeah. You need to work on the chemicals which are produced and identify the companion plants. It is very important.
Yes, sir.
Yes, sir.
Yeah.
Sir, would it be possible if you could send this paper, Khan et al. 2018, Outlook on Pest Management?
Okay, I can send it to you.
It's available online, ma'am. It's there.
It is available online.
Okay, sir.
It's a freely available paper, but if you want, you can, any paper you want, I can send it.
Okay, sir. Okay.
Yeah.
Excuse me, sir. I have one question. Is there any opportunity for students to have a collaborative research project with and IRI? Like PhD students, they can have the part of their research work there.
There are opportunities, but it has to be. You must have a scholarship. You see, here we have students from Africa who are funded through the German DAD. German DAD is a German government scholarship program. But what we have is only for African students. But we had some students who came from India who did some work, but they came with a scholarship, their own scholarship. So if you have a scholarship, you could be linked with a scientist who could supervise you jointly.
Thank you, sir.
Dr. Nikhil Raj has a question, but I think this has already been answered. What was the status of non-lepidopteran pest and sucking pest under push-pull based crop cultivation? Yeah, this question has been answered already. And does your institute provide funding on this topic? If some, no, our institute doesn't fund such things. But of course, you can submit your proposal to DST or DBT or other fund agencies. Or you can approach ICR for NASF funding if possible.
Yes.
Yeah.
Ma'am, Subbi ma'am has one more question.
There is a question in chat box, sir.
Subbi madam.
What should be the ideal plot size for doing push-pull study?
The ideal plot size is a plot from where you could have attractancy, enough attractancy. A 50 meter by 50 meter plot, that is a quarter of a hectare, is something which farmers plant. But you could see that how much is the attractancy, how far the trap plants can attract the pest. It depends on the pest to pest and crop to crop. But for the maize stem borer, we found that they can be attracted up to 50 meters.
Yes, Subhi, any more questions? Any more? Because she is working on maize. She is in Maize Institute of Maize Research. Any other person from Maize Research having any query?
No, I will.
Yes, ma'am. I have a
Thank you
question.
so much.
Yes, Anand.
Yes,
Yes, he is a student here. Yes, you
Yes.
can ask.
Good evening, sir. Here in India, it's a evening, while at your place, it's a good afternoon.
Yes.
I have a question. In maize ecosystem, ants plays a major role for controlling not major role, but some scientists have reported in Africa, they control fall armyworm. In 1990s, they have reported, 2 reports are there. So anybody working on ants, which is controlling fall armyworm or any other stem borer or pest in maize ecosystem in Africa?
Fall armyworm did not come in 1990s. It came, the first report was 2016
yes, sir.
in Nigeria, and then it moved to other parts of Africa. But there are many predators, earwigs and predators, which can control, and we have shown that in some of the papers for the stem borers that in nature, there is a large percentage of eggs are damaged by earwigs and other predators, ants. So it is very common. But when you plant desmodium in between maize, it provides the shade on the soil, and it also the predators also multiply there. So those predators also are very beneficial in the controlling the pest. So there have been reports, yes.
Thank you, sir. And even I heard that some farmers spray sugary solution to attract in the maize ecosystem.
Yes, they can put some sugary solution to attract the ants, so the ants can eat on the eggs of the some of the borers.
Yes, any more questions? Yes, anybody else wanting to ask?
Hello, ma'am. Can I ask one more question?
Definitely, Subhi.
Sir, happy to listen to you, and we have been reading you since long time. And I am working in Indian Institute of Maize Research. And from our AICRP and also from our own experience, and we are finding that this desmodium may not work in the wild agroecologies of India. And the main problem we are facing is the multiplication of the seed and also its growth. So sir, by seeing the chemical profile and seeing the close relation, any crop which is closely related to the desmodium, so we think we can work with the tephrosia. So what's your opinion on it?
You can work with tephrosia if tephrosia is useful, and it does not. If it can be planted in between the maize, you can use it. But I have not come across. Tephrosia has to cut down and put in the field to increase the fertility and improve the and control and improve the fertility. So you need to test whether it repels the stem borer and fall armyworm or it is not, because I have not used it, Tephrosia. So whatever is the best for the region, what is acceptable to farmers, you can do it. So please conduct the research and see that if Tephrosia is better, compare it, and if it is better, you just take it to the farmers. Yeah.
Thank you.
Yeah.
Yes. Anybody else? Okay, then we'll close the session. Thank you, sir, very much for being with us for such a long time. And as director, sir,
Yes, ma'am.
don't
Yes, ma'am.